And lastly, our next meeting will be on March 3rd, but now I would like to hand it over to Brandon to introduce our speaker for today. Thank you.
Awesome. Yeah, so it's my pleasure to introduce Eugene Katruka. He's a research scientist at the Utrecht University in Utrecht, Netherlands. I've been really following along with what he's been posting on Blue Sky with his big volume browser, so I'm quite excited to see that development happening. Largely, he works in areas of advanced microscopy, cell biology, and biophysical modeling, as well as image analysis. I think he's a self-described big fan of open data code, papers, and so I think most of his scientific output is openly accessible. Today, I think we're going to learn about his single molecule imaging using the LLS7. So Eugene, please take it away.
Thank you. Thank you for your kind introduction. One second. Let me just share my screen. Okay, everybody can hear me, right?
Yeah. Yes, we can
Great.
hear you.
So, yeah, today I'm going to be talking about single molecule and lattice light sheet imaging. But I'm going to start with the things that brought me into these two areas, which were basically relatively recent. And those are the biological structures that everybody likes. Well, I like it a lot. So these are microtubules. And you probably all know what are microtubules, especially since you heard about single molecule. This is like the holy grail. But I just wanted to jump some time back and say that actually they were observed like long time ago, right? Like almost 130 years ago. Because the function that they have, right? So they form the spindles, so these polymers, which participate in the mitosis, and they separate chromosomes into two dividing cells. So now we're going to jump back 130 years to the modern times and see how we can see this for a cheap contrast, how we can see this kind of structures with the modern microscopy. And this is a dividing cell, and these are microtubules, and you clearly see very peculiar 3D arrangement of the network. And the interesting part that this network is super dynamic. So it's, when the cell goes to interface, it basically really reforms itself. And then if you have a different cell type, and this is immune cell type, this is a T cell, then actually the network adapts and adjusts to the other shape of the cell. And here we see the neuronal cell. So this is all exam data, right? And in neurons, you have these dendrites and axons. So you have these bundles of microtubules because they serve as tracks for the cargo, so you can see where the molecules go. And this is apical airway epithelium, and you see that the transport direction is ingrained from top to bottom, right? So this is where the filtering. And now we're looking at cardiomyocyte, right? And here microtubules are all bent because, of course, it constantly contracts and bends, right? So as you can see, microtubule can take like microtubule network can take variety of different beautiful shapes and structures. And all underneath this, right, you have this kind of polymer made from dimers of the tubulin. And it's pretty interesting that it's a good, also very good subject for a super resolution microscopy because they're just 25 nanometers thick. Well, right, so we know the diffraction limit is almost 10 times larger. And the interesting part about them that they have a polar surface. So basically, the motors which transport intracellular cargo can recognize the direction of the surface, and actually specific motors like kinesins in red or dyneins in green, they move only in one specific direction on the scene. So the data that I showed before, right, those are actually expansion microscopy, really kind of nice recent technique, but it kind of basically shows you only the rods, right? So molecular microtubules being molecular rods, where they are inside the cell, but it doesn't show you the direction. And then if you actually look at this is the map of Manhattan, right? And this is one of the places which is known where you have like a lot of one-way streets. So if you want to travel, or if you want to see where the car goes, or how would you want to travel or deliver things, you not only need to know the direction, right? But you also not only need to know the position, but also the direction of the traffic. So to solve this problem, around probably now almost already 9 years ago in the labs in our department, we developed so-called motor paint approach, where what we do actually be basically permeabilize membrane of the cells and very gently fix them so that the molecular structure of the microtubules does not get damaged. So then the microtubules remain preserved, and then we add purified kinesin motors, which have some label on top of it, and they can diffuse inside the cell. And surprisingly, they still can walk along the microtubules. So if you now look at the recordings of how it looks like, it looks something like this, right? So as you can see, so these are labeled kinesins, which actually diffuse in the solution, then they bind, find microtubule, run along it, unbind, diffuse again. And since we have a lot of them, right, so it's basically like paint, but this is motor paint, right? So molecular motor paint. So now we can apply standard algorithms, right, to localize individual dots. And that will give us the position of the rods. But since we also can build now all these subsequent localization over time into the tracks, we can have a direction, right? And this is kind of, yeah, I don't know how to sell it. It's like this, I call them like microscopy friendly cells. So these are these 2D cells which are grown on a flat surface, right? So then it's very handy to image them with a standard illumination. And you can see that in this case, right, so we have this map and it's mostly radial. So the colors here correspond, right, to the direction of the movement. So you can see that in this case, so this is like the center of the cell, and on the top right, most of the traffic goes from the center, like how the purple arrow is showing. And the opposite is the sign, but there are some exceptions, right? So there are some purple microtubules inside the sign. So that's become really interesting. So how these microtubules, why they are flipped around. And this is right, so it's just most simple cell, like the most flat cell that we were looking in interface. So there is even kind of worse attraction to the flatness, because to reliably image individual molecules, usually people employ so-called TIRF microscopy, right? And TIRF microscopy basically illuminates very thin layer close to the cover slick, around 100 nanometers, which basically gives you kind of the optical slice, which is much thinner than kind of anything else, like much thinner than confocal, right? So then basically you remove all out of focus, all these diffusing molecules go out of focus, and you have like really bright single molecule signals. So this is very nice, but this is really not a kind of a good choice if you want to reconstruct 3D architecture of like really thick cells, and some of them can reach up to 20 microns, right, in the depth. So that's basically not suitable for volumetric imaging. That we kind of also realized, but luckily 1 year before the group of Eric Basic, right, introduced, well, this Lattice was introduced earlier, but then they published the Lattice Lightsheet Paint and Palm approach, where they basically imaged the whole cell in 3D using Lattice Lightsheet, using single molecule approach. I will call it single molecule, but I mean, yeah, paint or palm, to me, it's a little bit all just a matter how you make the molecules clean. So then, of course, kind of logical step was just to take the Lattice Lightsheet and apply it for the motor paint. And the main person in charge of this project was Malina. At that point, she was a PhD student in our lab. So actually, the majority of things were done by her, and I was just assisting in one or another things. And now, we basically, at that point, we have been, I think, the first user program for Lattice Lightsheet. So we got this instrument still kind of a little bit in the stage of the testing. But we decided, okay, let's just try and see if it not work, it's not working. That should be kind of relatively easy. So that was the idea, this was the protocol that we made. So basically, that's I already explained that we fix and permeabilize cells, then we add beads. I will explain why we did it later, but it's basically for point spread function calibration. And then, like in the same way as a regular model paint, you just add purified motors and then you image it. But now, of course, you do this under the angle. And the interesting part is that we chose the different approach that the Eric Basic group did, because in their in the data that I showed before, they basically continuously image volumes, right? So they constantly scan one after another, after another, after another. And then basically they can also, they have all the fiduciary in space and they can do drift correction and adjustments. So, and actually they have to, as I remember, I think for multicolor data set, they image something for a week, which is like super large and long. But in our case, we couldn't use this approach. Why? Because of course, like if we want to track individual kinesians, right? So if we do the volume, if we scan something like even like 30 frames with the lattice light sheet, when you scan, you come back, this motor couldn't bind or it already moved, so you kind of don't know what to do. So instead, what we did, we actually parked the light sheet in one specific position. We acquired some specific number of frames, in this case, for 1,000, right? And then we moved it and we acquired again, moved it, acquired again, moved it, and so on and so on. So to our delight, actually, it worked pretty well. So we can see individual molecules. That was really nice. And then basically now it doesn't matter if it's lattice light sheet or not lattice light sheet, you just can use any algorithm to detect individual molecules. And then for the Z-stack, we basically did the reconstruction of all the individual slices. And here you can see again, right? So this is the microtubules inside the cell. And then you can stack them together, and that gives really nice 3D picture. So we were really happy with it, but there was only one single problem, is that basically like each of the frames that we acquired, right? It was basically single slice, right? So it was really like 2D picture. So in each individual plot, so if you look at one specific, if you look just at very nice angle, it looks really nice. But as soon as you start to look from the back, you see that it's not really on its 3D data. And to solve this problem, well, I mean, it's also like not something that we invented, but usually like one of the easiest ideas behind that now, when you acquire the single molecule data, you basically want to encode the Z depth, right, in each individual slice. And this idea is to introduce cylindrical lengths, and that modifies point spread function. So if you have a point, right, in the focus, it looks like a round point. But then if you start to move in one direction, like from the focus, it becomes like horizontally stretched, and in other, it becomes vertically stretched. So then actually you don't need to modify that much the detection and the fitting of the molecules. And here what you can see is the width along the X and Y. So there are two beats, and you see that they are different depending on the defocus. So you can do it with the beats. And then basically you subtract one from another, and then you have a very simple function. So if you have these two bits, since this function is monotonous, right? So if you now have these two bits, you calculate their difference, and then you can go back and define actually the Z position, right? So basically from that point, you encode the Z position in the shape of the point spread function. So that was really, really nice. The only problem was that at that point, the microscope was in the warranty, and I was really excited to come and say like, yeah, we're just going to unscrew the camera and we're going to put cylindrical lens there. Like, what could be the problem? But somehow there was not that much enthusiasm from Zeiss, and they were not really sure because, yeah, I don't know, but maybe because just like the alignment, and so they didn't guarantee anything. They said like, yeah, okay, let's try something different, maybe less destructive. And the story is that there is already optical element inside the lattice light sheet which can modify penetrance function, and these are these Alvarez lenses or Alvarez plates, and the function of these plates is actually to correct for the different cover slip thickness. But what we found out that if we take a cover slip with just high precision, so right, so kind of probably most of you know that all objectives are optimized for 170. And then if you kind of go up and you put the wrong value, that actually points per function becomes astigmatic. So here you see the maximum intensity projection on different axes. And then it turns out that, yeah, if you carefully characterize all these things, you can find the value where you can build this function, and this function really kind of monotonous. So you can use it to map the width in X and Y of the point spread function into the defocus. And this is actually worked pretty well. If you look at it, the range is relatively large, so where the thing still works, right? So it's almost 2 microns, right? So then that means that you can take smaller steps out of that. So we were pretty happy with that. And then when we introduced this anti-correction of the cover slip, what we saw that we really happy to see that indeed we can see molecules of different shapes. And this is the example of the raw data. Then it had to be filtered from all the molecules like diffuse, the static molecules. So we perform some temporal median filtering here on the left at the bottom. And on the right, you can see like individual kinesin molecule, which is which has a raw data, and then we have a trajectory where the molecule moves. And from this trajectory, we really can decode X, Y, and Z. So having that in mind now, once we acquired single individual slice, instead of being absolutely flat, now it becomes like a small volume right around it. This is what is shown here, how where the color specifies the depth perpendicular to the slice. So that's basically the whole story behind the methods of the Lattice Lightsheet Model Paint, right? So you acquire all these data sets and then you perform localizations. Of course, once in a while the system drifts because in principle, it depends how long you need to acquire. In principle, I think we have different data sets. Some data sets really were around 1 day, but I think majority was something like 2-3 hours. So then you can correct for drift within single acquisition between different slices. You have all positions, and then if you perform tracking, you can actually assign the directionality of the microtubules. So what we did, we used these Urquhart cells because they're relatively small, and this was like the final result. This is actually one of the final figures in the paper. So this is the reconstruction of the microtubule network of the full Urquhart cell. And of course, this is just the rods, right? But now this is the rods already with the orientation. So as you can see now, we need to map the orientation not only in the 2 dimension, but also in the 3 dimension. And actually, you can see maybe in the beginning there is a centrosome, and then like in all colors, the tubules go around it, which means that it is we kind of know that it's supposed to be like this. And of course, it's light sheet, right? So then you just can scan for longer and longer and longer and longer. And in this case, we have like the whole set of Urquhart cells. And this is just a single acquisition. This acquisition was, I think, around 7 hours. And then there was a little bit of analysis. But in principle, it worked really well. So we were really happy to see that the method works also in 3D. So that was when the paper was published, now 2 years ago, right? And it's open access, and I provided the PubMed ID for that. So now it's already like 1 year since, so it's time a little bit for reflection and the critical look at that. And usually nobody is better at critical look than the authors because they know what is kind of not working or what was working. So one worry that we had that how much reproducible this method is. So we kind of assuring the demand, but because right, we kind of the way we introduced this point spread function engineering was somehow kind of like arbitrary, right? So we didn't do kind of rational design. We did it, and then we saw like, oh, okay, it works. And the story is that actually, if you use thinner cover slips, for example, number 1, right, which have like around 150 micron, and you cannot introduce the aberration, right, because you're already kind of below the thing. So it doesn't matter how you correct for the cover slip, your point function remains kind of like still pretty round. So that's why we were really happy when last year there was a paper published, and it was cited also our work from Marcus Sauer group, where they basically used our method not for motor paint, but for DNA paint, and they developed their own DNA paint. techniques, and I'm showing just the first figure from the paper because it has microtubules, but it's just a proof of principle. And in principle, this is the whole story about immune system, immune cells and receptors and the antibodies. So you should check the rest of the story. But I was just very pleased that our approach and even our analysis methods were robust and were reproduced. So this is not, in my opinion, it's not always, not like off, not always, but let's say not often it happens. I think that it's really interesting and kind of really to use technique for commercial setups. And the other problem that we had was, of course, that we had to do this laborious point spread function calibration. That's why we added the beads. And then we had to scan the volume multiple times, detect the beads, which is not really very kind of straightforward procedure when you try to do it in 3D, plus a lot of kinesins floating around. So yeah, that was also like something that we were thinking going to be not really easy for other people to use it. But recently I discovered that it turns out there is a point spread function wizard in Zen. I don't know when it appeared. Maybe Houston can help me. But yeah, so the story is that in principle you can just scan it, and it has very user-friendly wizard where you can pick different beads, and then it basically distills the point spread function. So it will pick like beads, crop the stack, so you can then even like fine-tune them. So if you haven't seen it, you should try it. And still, even with this thing, right? So this kind of story about this point spread function was worrying us a little bit. And the interesting story that after some point, I think after a couple of years, we received the serial. So we were in this first user program where we had this experimental microscope, and then at some point it was shipped to Zeiss, and they shipped us the new one. And in the new one, the worst thing that there was the astigmatism was much worse. I mean, and when I say worse, that it was much less, so the point spread function was much better. So at that point, Lucas, who is like the head of the lab, was really disappointed. He's like, "Yeah, you need to send us this other machine, which was like really bad. You don't want to have good point spread function." But at that point, that was already a little bit later, right? And interestingly, there was the whole set of papers during the last couple of years, which actually eliminated all the stories kind of with astigmatism and point spread function and things like this. And I think probably the very first one was this paper shown here from Jonas Rees' group. And basically, the story about these papers is that it's not necessarily that you need to acquire these stacks of the bits and then try to distill point spread function. So what you can do instead, you can actually record your blink and single molecule data and then apply very sophisticated optimization routines, which from all this array of blinking data will try to extract the point spread function. So that's kind of what they did in this paper. So this kind of inverse modeling of point spread function. So I think they used TensorFlow to just like rebuild all the things. And of course, all these papers, they also take in mind that in point spread function cannot take any like arbitrary shape, right? So in principle, there is a very good theory behind it, right? So then you can use Zernike polynomial, so your number of actual parameters reduces pretty efficiently. So then all these brute force optimization systems, they can really give you a really good solution. So there is another paper from Netherlands, from Schuursteling and Bernd Rieger lab. So this is in Delft, where they actually also use the blinking data to extract point spread function. And there are also a couple of papers that use, of course, the neural network vision there, and also optimization stories. So that's from Joris Schepman. And there is also a very recent preprint from Yimin Li and Shanfu. Actually, these papers, all of them, they have the code available. And I think all of these groups usually, I mean, I don't know, and not that I don't know any. I mean, I'm not sure, but usually you can contact them, and if you have the data, you can actually uh, do a pretty good collaboration. Because in principle, all this analysis is relatively complicated and computationally heavy. So if you don't have infrastructure in your labs, it's probably going to be not very easy to set it up from the one point of view. One other point of view could be a good PhD or postdoc project. But what I want to say is that having these tools available actually removes the need to have a very specific stigmatic points per function. So all you need to have is you need to have points per function which is asymmetric in Z, and then all these tools kind of are going to reconstruct it from the blinking data. So you don't even need to add bits and acquire these things. So this is, I think, it's pretty fantastic. And we actually tried to reanalyze our data with the group, with the Emily group, and that actually looked quite okay. So this is like the preview, the volumetric data. This was our analysis that I described before with our plugins and processes. But then if you put it to the system, right, which doesn't know points per function, and it learns points per function from that, and it localizes the thing. And I think it looks pretty good. In my opinion, it's much more crisper and fine than our data. So there will be soon, I think it's not in the preprint, but I think it should be soon somewhere published. So there, all the details will be there. But the code is already available, so you can already just download and run it. So they should. A couple of kind of final remarks. So yeah, you have to deal with the drift because this is a really long-term acquisition, and individual plane drifts, usually there's a lot of algorithms to correct for that, based on like partial reconstruction and then cross correlation. But then the story is that you will also have 3D drift correction, and in case of lattice light sheet, where it drifts, because if it drifts in Z, it actually also drifts in Y. So I really put a lot of my head around this thing. So it's really not that trivial. But again, in the recent latest light sheet system, I think in like recent Zen at some point, the reflection focus was implemented, right? So you can use reflection from the cover slip to kind of go back to the Z that you chose in the beginning. And that really helps. So at least in this case, you know, your sample will not drift away. Like, so this is one of the most scary stories. So you, after 2 hours of acquisition, you're looking that your sample gradually goes out of the foo, and then you realize that, okay, everything is needs to be started again. So that, I think, a really nice feature. And that part is actually, so we also share the code and everything for the paper and analysis. And there is a macro which makes possible all these acquisitions. I didn't mention it. So right now there is no this store focus, recall focus function, but I guess you can just add it and use these things. The other thing, well, okay, I'm jumping a little bit, but that's fine. The analysis is relatively complicated. And because, well, I mean, the fitting is okay, but then again, you need to DSQ all these things. And here I'm going to preach about the big volume browser. This is the plugin that Brandon mentioned. This is a plugin that we developed in our lab, and in principle, it was made to work with really large terabyte volumes, but also in the end, it also can render single molecule data meshes. And then you can do a lot of different transforms. And one of the transform, it doesn't matter what kind of object it is, it's a volume, a smell data, or meshes. And one of the transform is actually this Q. So basically, I really recommend the plugin to use it a lot because then you don't need to, right? So the story is that usually you acquire something on Lattice Lightsheet and then you need to wait for this queue, and then it generates the file. If you want to have cover slip transform, right? This file will be also like 2-3 times larger. And then you just want to check quickly, you know, if something goes out of focus or if like the event that you tried to capture didn't happen or not. And especially in 3D, it's not always trivial. So this plugin basically you can open the raw CCL file, drop to it, and then just like draw a slider, and you will have like the whole view. It's first time, and in principle, it can also work with the data which is larger than your GPU size. Basically, visit it and check it out. If you have any problem setting it up, also let me know. And last point on the critics list is that the custom-made probe, indeed, we had to purify the Keynesian, and well, there's kind of no updates about that. We don't plan to make a company selling this, but in principle, the protocol is described. But if you are not that big fan of microtubules, as I am, for example, or you want to use something else, in principle, you can use DNA Paint, right? And that works really well. You can use Storm anyway you make your probe link. That's like, should be fine. It's not such a big deal. And I think modern Lattice Lightsheet even come with the two cameras, so you can do simultaneous recording. But one of the most lazy things, and I really like lazy science and simple things that work, that I discovered recently and actually tried just like before, like a couple of weeks ago, was the preprint from a couple of groups where they actually found this is one of Janilia dyes basically, which blinks on its own, and it blinks in the water solution. So the cool part about it is that if you ever, so we did Storm from probably from 2017. And this first protocol, you have to do this oxygen scavenging buffer, and then you need to add this mare, which smells like terribly. And then if something is, yeah, so it's like really complicated, like to make things blink. And this one is just like given. The only problem is just one color. And there are commercial, it's commercially available, fused to nanobodies, which is also pretty neat because then you just incubate with 5 minutes you incubate with the primary staining, and you can go and just image this. So this is an example of the raw data. Of course, you probably already guessed what kind of structure is stained to microtubules. And this is basically the preview. So this is pre-scan in one color in green, and this is the analysis results of the blinking dye, which are very quick and dirty. I didn't do the full pipeline. I just wanted to see like, okay, do I see something or not? And it seems to be working pretty well. So, and this video and almost like 90% of all these videos were made in the big volume browser. And again, so this dataset was also just acquired and then it was queued inside. So I didn't have to write any like affine transforms for the raw data. I'm pretty happy with that. It makes things much faster. So I think that's pretty much everything I wanted to tell you about, and I'm going to finish with the acknowledgements. Again, kudos to the first author, Melina, and this whole work was done in the labs of Lucas Kapitan and Anna Achmanova, together with a lot of talented people which were solving hard biological questions, but once in a while bring very interesting sample for us to image and play with. And thank you for your attention. That's
Excellent. Thank you so much, Eugene. That was great. I think we have like 20 minutes or so. That's plenty of perfect timing for questions. I think Eugene did a great job of saving a lot of time for questions and answers. So I would open it up to people on the floor. Feel free to turn your camera on if you would like to and ask questions. That's probably the easiest way. You can raise your hand first if you'd like. I think that would be ideal.
I have a question just about the image analysis. I think you explained it really well, but our system is in a core facility and we've really wanted to try to do as much as we can within the Zen software. Have you had any, do you have any ability, like depending on the data size or anything like that, to do any of the deskewing or segmentation in that in the Zen software?
Not that I'm aware of. I mean, you can. You mean desqueuing on the fly? I think it's not possible. You need to do like an extra uh, new file, right? Which would
Yeah,
be
I think
large.
even just like if like can you take the data and deskew it and work with it in Zen or do you have to take it out and use these third-party platforms?
Yeah, but all this it's a Fiji plugin, right? So you can just install it on the same PC where you have your Zen, and there's no limit in the licenses and things like that. And uh, I think we use it. We are imaging facility, right? And almost in all our PCs together with uh, kind of the manufacturer software. We also usually have Fiji, so for quickly a look. And now there is also this 3D plugin, so then we can just also in parallel, while the Zen is busy acquiring something, then you can just very quickly look at the data in 3D and maybe make a movie and show it on the next meeting, which not always possible. You know, if you have only one Zen license, so then you use it to acquisition, and then you need to also maybe book some slots to do the skewing, and then maybe you can do like only some movies after that. So that actually helps in our workflow for Lattice Lightsheet. So it really speeds up things, especially for people like uh, the newcomers, you know, like the master students, the people that not stuck with the old things to do. So I don't see why it's right, the Fiji and these plugins, it's not something which is like competitive. It's more just complementarily parallel that you can use these things. So,
Okay, thank you.
but I agree. Well, I mean, I saw Kirsten is here, so I agree that it would be nice to have possibility to kind of at least look at the data on the fly. So.
Other questions? I would say I'm after seeing what you were showing with the live deskewing, I'm getting the plugin as we speak. So I'm super excited to give that a shot, and because it looks like it's super fast as well, right? I mean, I think you're doing a great job on that. So I'm very excited to use it.
Thank you. So yeah, but the story is that also I think what one missing thing right now, but I'm working on that, is that it doesn't have like the way to export animation yet, right? So it's usually what people, right, use, for example, in Iris, right? You acquire a lot of data, then you quickly do a video, and then you can like bring it to PI and say like, okay, so is it a good data? What we're gonna do? Are we gonna do that experiment or that, right? So once you look at the data, you kind of realize what is happening. But that's gonna appear in a couple of months, I think. It's in the plans. So, and then the only story could be is the best way to kind of go around the large data and the resources that you have. Uh, so, I mean, it's not any secret or anything, but uh, it really helps to if you get stuck somewhere, you can just write it as yeah, you can write a personal email, or there are like it's on the forum or on GitHub or whatever. So because it also helps me if I see where people struggle, then I will write documentation better. So that kind of feedback I really appreciate. Yeah. But it is a big problem, yeah, with the modern cameras which increased in size and expansion methods and all the things, it really becomes pretty challenging to work.
Eugene, if you want to stop sharing your slides so we can see people.
Yeah.
Oh, thank you. Great. Not a question, but there is a comment in the chat from Alex. I don't know if you want to expand on that, or I can read it as well.
There are a lot of solutions for point spread function in Hugging software. Yeah, what can I say? There are some solutions. So there is a point spread function distiller. Yeah, it's the convolution software for people that don't know it. But it's a commercial software which is not very cheap. So if your lab has money, of course, you can use it. And we also use it for the convolution. It's very easy to use, but is there a workflow to do Lattice Lightsheet localization? I'm pretty sure there are not. We know them very well. They're actually not so far from us. I think it's 15 minutes by train, and we cooperate we even have some joint master students. So I'm not sure that there is like solutions on the website. If you can just go to the website and upload there like 3 terabytes of data, I'm pretty sure that if I understood the question correctly, but you know, I really think that with this self-linking story, actually now you can breach the expansion and single molecule imaging, for example, because before it was kind of a little bit challenging to do because you have to add these specific buffers, right? And this self-linking, but this is actually also what the authors say. But then to acquire expanded sample, right? Expanded samples are huge. So where do you go, right? So then you will go to Lattice Lightsheet. So I think that's worth a try. Because in principle, kind of expansion kind of dominates the volumetric kind of high resolution imaging. Because of course it's pretty easy. You don't need to do all this tedious analysis, molecule localization, and things like that. But it has its own downsides, right? So in the sense that like you have very low preservation of there are a lot of your antibodies and dyes, they just get flown away. So your labeling density decreases a lot. For example, I haven't seen the holy grail of the localization microscopy, a hollow microtubule, right? So if the microtubule is hollow, that means you're like on the next level. But not to my knowledge, maybe I haven't seen this in expansion. Or maybe, oh no, like I can't remember this like that very clear image, but maybe there is, yeah.
Okay. Well, I'm not seeing any other questions. So with that, maybe we'll end the meeting. And thank you again, Eugene, for a great presentation today. And thank you for being with us.
Thank
Thanks
you,
for having me.
Eugene.
Yeah, thank you. It was great.
Yeah, I hope to see everybody. What is it, March 3rd?
March 3rd. Yeah, that's our
Sure.
next meeting. Yep.
All right. Excellent. Thank you all.
All right, bye-bye.
Thank you.